Hello friends, welcome to Sam Biology class. Today we shall discuss about the mechanism of action of hormones. The first step in the action of hormones is the formation of hormone receptor complex. Hormones are extracellular signaling molecules. Receptors are specialized proteins. For some hormones, such as proteins and peptides, receptors are located on cell surface, whereas for others, such as steroid and thyroid hormones, they are intracellular, found either in the cytoplasm or in the nucleus. In some cases, after binding with the cell surface receptor, the hormone receptor complex leads to the generation of intracellular signaling molecules known as second messengers. Examples of second messengers are cyclic nucleotides such as cyclic adenosine monophosphate and cyclic guanosine monophosphate. Other second messengers are inositol triphosphate, diacylglycerol and others. Whereas in case of steroid and thyroid hormones, the receptors are intracellular. Such hormones, after diffusing into the cell, binds with receptors either in the cytoplasm or in the nucleus. The hormone receptor complex then binds with specific DNA sequence and activate the transcription of specific mRNA, which are then translated into proteins. These proteins bring about definite changes in the physiology and behavior. For other hormones, for example insulin, the receptor is also located on cell surface. But here the hormone receptor complex itself brings about the final action of the hormone. So some hormones act through second messengers, others act through protein synthesis, and remaining hormones act through the hormone receptor complex itself. One of the means by which hormones exert intracellular action is to stimulate the formation of intracellular signaling molecules known as second messengers. In addition to cyclic AMP and cyclic GMP, two other important second messengers are calcium ions and associated calmodulin and the products of plasma membrane phospholipid breakdown. Here we shall discuss about adenylate cyclase CAMP second messenger system. Cyclic AMP is the cyclic form of adenosine monophosphate. Cyclic AMP is produced from ATP by the action of enzyme adenylate cyclase. Adenylate cyclase is a membrane-bound enzyme located in the inner side of the cell membrane. Adenylate cyclase is activated by a membrane-bound regulatory protein known as G protein. There are different forms of G protein. The one that activates adenylate cyclase is known as GS protein that binds GTP for activation. GS protein is attached to the transmembrane receptor. When a specific hormone binds to a specific transmembrane receptor, G protein is activated, which in turn activates adenylate cyclase. Activated adenylate cyclase then catalyzes the formation of cyclic adenosine monophosphate from ATP. Cyclic adenosine monophosphate CMP is an important second messenger system. Cyclic AMP then activates protein kinases, in particular protein kinase A. These activated protein kinases catalyzes the phosphorylation of various intracellular proteins. These phosphorylated proteins are responsible for the final response. When the action is over, cyclic AMP is converted into inactive 5-adenosine monophosphate by the enzyme phosphodiesterase. This terminates the hormone's action. In contrast to protein and peptide hormones, other hormones including adrenal and gonadal hormones, thyroid hormones, retinoid hormones and vitamin D binds with intracellular receptors. These hormones act through protein synthesis. Being lipid soluble, such hormones easily diffuse into the cell and binds with intracellular receptors. The receptors for such hormones are located in the cytoplasm and in the nucleus. If the receptor is cytoplasm, then hormone after diffusing into the cell binds with the receptor in the cytoplasm and form hormone receptor complex, which then diffuse into the nucleus through the nuclear pore complex. But if the receptor is located in the nucleus, then the hormones after diffusing into the cell move into the nucleus through the nuclear pore complex and binds with receptor. In both cases, after its formation, hormone receptor complex binds with specific regulatory sequence of DNA known as hormone response element. This binding activates or repress the transcription of specific genes and formation of mRNA. So, minutes, hours or even days after a hormone has entered a cell, new proteins appear which then control new or altered cellular function. Naturally, this process requires more time than the second messenger-mediated response. Many hormones such as fibroblast growth factor, growth hormone, insulin and insulin-like growth factor 1, leptin and prolactin use enzyme-linked receptors for intracellular signaling. Such receptors, when activated, function directly as enzyme or are closely associated with enzyme that they activate. Such enzyme-linked receptors are proteins that pass through the cell membrane only once. These receptors have extracellular part which binds with the hormone, and the intracellular part have the catalytic site. When the hormone binds to the extracellular part, an enzyme immediately in the cell membrane is activated or occasionally inactivated. Although many enzyme-linked receptors have intrinsic enzymatic activity, others rely on the activity of enzymes that are closely associated with them. All such hormones use the enzyme-linked tyrosine kinase receptor for cell signaling. This figure outlines the mechanism of action of leptin. Leptin is a hormone secreted by fat cells. This hormone is involved in the regulation of appetite and in energy balance. Leptin receptor exists as a dimer. Binding of leptin induces conformational changes in the receptor, which in turn phosphorylate and activate intracellular associated Janus kinase 2 molecules. Activated Janus kinase 2 then phosphorylate other tyrosine molecules that are located in the receptor JK2 complex in order to mediate intracellular signaling. Intracellular signals include activation of signal transducer and activator of transcription protein, which in turn activate the transcription of leptin target genes and initiation of protein synthesis. Phosphorylated Janus kinase 2 also activate other intracellular enzymatic pathways such as mitogen activated protein kinase and phosphatidylinositol 3 kinase. So friends, this is all about the mechanism of hormone action. For more updates, subscribe my YouTube channel, Biology Class.